All right. Welcome, everybody, to Standing for Truth. My name is Donnie Badinsky, and it is a privilege to be your host and moderator. Tonight, we're bringing you a highly technical and important debate on genetics, mutations, and evolutionary theory. The debate resolution is this: are mutational effects a problem for evolution? Representing the affirmative position is Paul Price, with Dr. Zach B. Hancock arguing the negative. This should be a fascinating and in depth discussion. And so let's get right into it by breaking the ice and getting to know you gentlemen a little bit before our debate showdown begins. I'm pumped for this. I'm excited to see how you both are going to argue, what points you're both going to bring up, and how you guys counter those. I think this is going to be good and thought provoking, that's for sure. So, Paul, why don't we start with you? You are in the affirmative here tonight. So, a brief introduction, not your first time here on Standing for Truth, nor is it your first formal debate on a topic related to origin. So, Paul, how have you been? But also tell us a little bit about yourself.
Well, Donnie, thank you as always for having me here. It's always a blast and an honor. And I have been busy. This debate has actually been 8 months in the preparation and the making, believe it or not. And I think I actually needed all 8 of those months to actually get anywhere close to uh, Dr. Hancock's level of being able to have an intelligent conversation on this. So I'm greatly indebted, I want to say, to Dr. Hancock for being willing to engage in this debate, which I have to say is a rarity today among his peers. There are few that would be willing to do this. And in my mind, that makes him a much better scientist than those other ones that are unwilling to dialogue at all. And I want to say this too. I'm not gonna pull any punches in this debate tonight, but it's all in love, not hatred. So I want to get that said in advance. As far as who I am, for those that aren't familiar with me, I have had a history of working in the field of creation apologetics. For 6 years, I was blessed to be able to do that full time professionally. Today, I work in the corporate world and I get to do this more as a hobby, but as also something that I would consider to be a passion. And I think it's a very, very important topic. So again, super excited to do this, very blessed and excited to be here. So with that, I will turn it over to Zach.
Paul, thank you very much for that introduction, helping us to get to know you a little bit. I do have your YouTube channel and links, relevant links where people can see more from you in terms of articles and talks that you've given. So very good. Dr. Zach B. Hancock, not your first time here either. The last time you were here was also for a formal debate, if I remember correctly, going back last year with Dr. Jerry Bergman. Time flies by when you're having fun. I know you've been busy, so I do appreciate the time that you've offered for tonight's very important debate on a technical topic that I'm excited to get into. So, Dr. Hancock, how have you been? And also tell us a little bit about yourself.
Yeah. So hello, everyone. My name's Zach. I'm an evolutionary biologist, currently a professor at Augusta University. The one of the reasons this has taken 8 months is because I just moved here and just started this job, and I have to tell you, if you've never been in academia before, this is way more work than I've ever had to do in my life. It's just like so much work. So I apologize to both Donnie and Paul for taking this long for us to be able to schedule this. This is the reason why it took so long. It's like trying to get used to a whole new life has been a real challenge. But I definitely appreciate Paul, what you said, and like you're having a bunch more time to do this. I knew that you were going to dedicate the time and research necessary to have a good conversation, and that's why I agreed to do this with you, because I knew you would put in the work to have the good conversation. So yeah, I'm really excited for this. I've been looking forward to it, and I hope that we get to talk some interesting stuff tonight.
Agreed. I like the fact that you both have put a lot of work into studying this topic. Of course, Dr. Zach B. Hancock, you and Dr. Dan Stern Cardinal published somewhat recently in a technical paper very much related to tonight's debate question on anybody just joining us. The question, are mutational effects a problem for evolution? For those that want to see more from Dr. Zach B. Hancock, please check out the description box of this video, but also here in the live chat. Thank you very much, Doki Doki. A link to his YouTube channel and another relevant link to see his technical work. So very good, gentlemen. Let's make sure we're all on the same page, and then we'll jump into our opening statement. Our first opening statement, which will be an affirmative constructive of 15. That'll be Paul Price taking the affirmative tonight. Then we jump into fast-paced debate here. So we jump right into a cross-exam of 5 minutes. Then negative constructive plus first negative rebuttal, 18 minutes. That'll be Dr. Zach B. Hancock. Then another cross-exam of 5 minutes. That time Paul leading the way, of course. And then the first affirmative rebuttal, that'll be Paul, 8 minutes. Negative rebuttal, 10 minutes. And then we'll wrap it up with a second affirmative rebuttal of 5 minutes. And then this is where we get you guys in the audience involved. We'll be having, we're pretty flexible tonight on the audience Q&A, roughly 45 minutes to keep about a 2-hour debate. So please, if you do have questions, challenges, objections, perhaps you hear something tonight that our debaters argue or put forth that you disagree with and you object to and you want to challenge it, tag me at Donnie or at Standing for Truth. That way I don't miss your questions. And we will have some fun interacting with your questions. I do enjoy the audience Q&A periods because I think they help us to expand even more on these topics, giving you guys a comprehensive debate. Then Dr. Dan Stern-Cardinal from the live chat, Creation Myths, good to see you. He will be having an after show over at his place, his channel. So do make sure to tune in for that, as I'm sure the conversation will continue for at least a couple more hours after this. Okay, I think we're on the same page and we're good to go. Paul, you just let me know when we're ready. And first affirmative constructive, 15 minutes. The floor is yours.
All right. We should be up and running. Do we see the slides, Donnie?
Yeah. Well, it says...
we're booting.
Yeah.
There we go.
There we go.
All right. So, Donnie, just go ahead and start. Let me know when you're starting my timer.
Starting now. Go ahead.
All right. Thanks, everyone. In tonight's debate, we're going to be discussing the extremely important topic, are mutational effects a problem for evolution? And my answer is yes, big time. But what do I mean in this debate by evolution? What I mean is the neo-Darwinian idea of naturalistic, universal, common descent. And this problem has been acknowledged in the literature. Back in 1995, Kondrashov realized that realistic parameters applied to well-established population genetics theory predict unstoppable fitness decline due to the gradual buildup of very slightly deleterious mutations, or VSDMs. He called this the stochastic mutation load paradox. He wrote, because the stochastic mutation load paradox appears real, it requires a resolution. Now, later, Dr. John Sanford would write his introductory book to a lay audience called Genetic Entropy and the Mystery of the Genome. And he would expand upon these ideas, but this here really is the core of Dr. John Sanford's genetic entropy. So please don't let anyone tell you that genetic entropy isn't found in the literature. It certainly is, just not under that specific name. In fact, it is continuing to be acknowledged to the present day. For example, we have out of the University of Arizona, the Mazel Lab. And on their website, they write, in the title of a 1995 paper, Alexei Kondrashov posed the puzzle, why have we not died 100 times over? We still don't have an answer. Most work on mutation load periodically renormalizes relative fitness to deal with the fact that fitness keeps declining no matter what, or else treats only a subset of the genome. So what is it that's causing this decline? Well, here we have a distribution of fitness effects, or a DFE. And this is basically giving us the spectrum of values of the fitness effect of all the mutations affecting the population. And what you see in the middle there in that shaded region is called the no selection zone. That is simply a way to understand that past a certain point, as mutations get smaller in their effects, natural selection is no longer able to see those mutations, can't act on them. And we call that cutoff point the drift barrier. And the drift barrier is a function of effective population size. We'll do more on that later. Dr. Michael Lynch is a well-established expert in the field of population genetics, and he states, for an equilibrium or well-adapted population, the number of beneficial mutations fixing each generation must equal the number of deleterious mutations fixing. So what that means is basically we need balance in our DFE, and we don't have that balance. As you saw, the DFE was highly asymmetrical. And what does he mean by fixing, by the way? What he means by fixing is new mutations that enter the population have one of only two different possible end results. One, they could be lost and be completely eliminated from the population over time, or two, they could become fixed, which means that they move to 100% frequency shared by all members of the population homozygously. Now, my opponent has actually recognized the importance of these DFEs, and he stated, you didn't even need to do the simulations. When you give the sorts of distributions they give, you already know that fitness is going to decline. And by the way, he means when he says they, he's talking about Baziner and Sanford. But this is a huge admission on the part of my opponent. But why isn't the DFE balanced? Well, if you think about it, it should be pretty obvious why it's so asymmetric. But here's Dr. Philip Garish. He states, even the simplest of living organisms are highly complex. Mutations, indiscriminate alterations of such complexity are much more likely to be harmful than beneficial. And Dr. Lynch himself made it even simpler by saying, it's a lot easier to break a gene than make a gene. And this here is why our DFE is so unbalanced. But that lack of balance under neutrality is going to lead to what I call the fixation ratchet. Dr. Lynch states, under neutrality, the cumulative rate of fixation of neutral mutations is equal to the per generation mutation rate independent of the population size. Now, it turns out that the vast majority of mutations will be effectively neutral in large multicellular eukaryotes, or LMEs, as I'll call them. Now, what this means is that each generation, regardless of population size, on average, you can set aside one individual's worth of new effectively neutral mutations as destined to fix. That means they will gradually increase in frequency over time, eventually to 100% theoretically. And so that's what I'm showing here with this red double helix. The blue double helixes represent deleterious mutations segregating in the load that will eventually be lost to drift. And so what we see each generation is that small percentage that are headed to fixation or en route to fixation, on average, grows by a little bit over time. That's the fixation ratchet, and that is what makes genetic entropy, or GE, inevitable. So here's how GE actually works in LMEs. 1, growth happens exponentially until a carrying capacity is reached or nearly reached. 2, total segregating mutational load continues growing until mutation drift equilibrium is reached. That's the point at which the number of mutations lost per generation equals or approximates the number being added. 3, even though the total segregating load reaches a plateau, the percent of the load destined for fixation grows each generation on average after the population stabilizes. That means that the average individual has a growing share of that total segregating load. And lastly, population fitness declines linearly. So I have set up a simulation in SLIM, which is the same program that my opponent, Dr. Hancock, used. And this is showing that initial population growth up to carrying capacity, followed by the gradual decline due to the buildup of VSDMs. Here you can see a view of the segregating mutation load. This is the mutation drift equilibrium I mentioned. It grows up to a plateau, and the height of that plateau is going to be a factor of your total population. And here's perhaps one of the most important slides that you're going to see in this whole debate. This shows you the percent of the load en route to fixation. And you can see after the population initially stabilizes, this percentage just keeps going up and up. This is the mean deleterious allele frequency. This is showing us that the average individual is starting to harbor a greater share of the total segregating load. And lastly, you can see that fitness declines, and you can extrapolate that. I ran this simulation as long as I could, but it's rather computationally heavy. So even after weeks on end, this is as far as I could get, but you can certainly see where this is headed. And we're extrapolating an estimated max value of a little over 40,000 years, assuming 20 years per generation. So about 2,000 generations here. Now, how has my opponent, Dr. Hancock, attempted to refute GE? The first thing we can say is that my opponent, over the past few years, has released quite a few videos and made quite a few different arguments in an attempt to refute GE. So at the very least, what we can say is this seems to imply that he considers this a serious challenge to his worldview, which it certainly is. But what we're about to see is that my opponent's attempts to refute GE with math simply do not work. So I'm going to distill this down here to the top 3 arguments that my opponent has made. 1, Hancock claims that the current human effective population size is 7 billion, and thus this whole thing becomes a non-issue because almost no mutations are going to be effectively neutral at that size. He tries to downplay the consensus figure of 10,000. But his problem here is that even the highest published estimates of recent human effective population sizes come nowhere near his figure. So at the very least, he would need to give us some kind of citation to back this claim up, which to my knowledge so far he has not done. But it gets worse from there, because even granting this figure, it is irrelevant, because his claim that we should not be concerned about the long-term history of humanity is patently false. After all, we're debating about the long-term plausibility of the theory of evolution. So to determine that, we would certainly be interested in the long-term effective population size. But you don't need to take my word for that, because if you go to Kondrashov's paper, he specifies we are talking about the long-term effective population size. That's what N sub E represents. We are certainly not talking about merely the current or recent or variance effective population size. Number 2, Hancock claims that his published simulation on SLIM invalidates GE. His simulation parameters are very loosely based on a single empirical study of a total of 48 artificially induced mutations in RNA viruses. Are we really supposed to believe this is representative of the broad human DFE? But even then, his simulation fails to replicate that empirical one that he cited in any meaningful way. The only aspect he seems to have taken from the empirical study is the average mutational effect near 50%. But he did ignore that in the DFE they showed, nearly 30% were neutral and nearly 40% were lethal. So if you average out neutral and lethal, you're going to get about 50%, yes. But nobody thinks that that's what the human DFE looks like. Hancock is cherry picking the data and misapplying them. His sim population also fails to stop growing at the carrying capacity that he set when you run it longer than the published number of generations. How was this not a red flag? The fitness scaling that he has in his code places downward pressure on the population, and yet it keeps going and going ad infinitum. What this shows us, by the way, here is his published results, stopping at 50,000 generations. But here's how it looks if you allow that same simulation to just keep going past that point. You can see it is absolutely not stopping at the carrying capacity of 1,000, and it just keeps going. This shows us that it's greatly, greatly unbalanced in favor of the beneficial mutations on his DFE. Also, at the effective population size of 1,000 in his sim, Hancock and Stern-Cardinal's deleterious DFE contains only about 2.5% effectively neutral mutations. Now compare that to Kimura's distribution, which has 52% at this level. So his DFE is 97.5% selectable. No wonder he got the result that he did. Everything about Hancock's simulation parameters seems to be engineered to maximize natural selection in ways that are entirely unrealistic, and there does not appear to be any consistent scaling logic applied. Fixing nearly any combination of his broken parameters results in fitness decline, just like the Maisel Labs said it would. Here I'm putting up his parameters juxtaposed with empirical human parameters, and what you can see is that there's simply no consistent ratio, no scaling, and many of his parameters are extremely far off the mark. Number three, and lastly, Hancock attempts to hand-wave away the genetic load model entirely, saying, when population geneticists use it, we don't take it as gospel. But at the same time, in an obvious demonstration of doublespeak, he says he wants to be clear that he isn't saying the model isn't useful. So, okay, if pop gen experts don't take it as gospel, why did Kondrashov identify this as a paradox that needed resolution? And why did Hancock himself bother doing a simulation based upon these theoretical principles and claiming his simulation invalidated GE? Dr. Lynch has choice words for this kind of hand-waving. He states, this is why we use population genetics theory to understand the process of evolution. We have a real problem in evolutionary biology with arm waving, way too much arm waving. The principles of population genetics are so well established that the credibility of any proposed scenario for any feature of evolution must remain in doubt until it can be shown to be theoretically feasible. So this is what this concludes for us. 1, neo-Darwinian evolution is false. Complex life is irreversibly degenerating. And 2, life, and therefore the earth, is young. This process happens far too quickly to accommodate deep time. This represents yet another independent contradiction of radiometric dating methods. Thank you. The SLIM files are available publicly here.
Paul Price, thank you very much for that first affirmative constructive of 15 minutes. To the audience, I appreciate the energy. Thank you for the engagement and interaction so far. Once we're done with our main sessions for tonight's formal debate, we will be jumping into an audience Q&A period. So reminder, if you do have a challenge or an objection or even something of clarification, send those in, tag me, I'll save them, and we'll respond to those in the Q&A. Okay, we're jumping into the first cross exam of 5 minutes with Dr. Zach B. Hancock leading the way. Starting, firstly, Dr. Hancock, just make sure to unmute before you speak, and then I'll start the timer on your first question. Go ahead.
Yeah, so can you just quickly share my screen?
Yes.
Thank you. Okay, so Paul, is my understanding, or is your understanding of effective neutrality that selection cannot act? That seems to be the way that you presented it in your presentation, that if it's effectively neutral, selection can't act on it. Is that true?
Well, the drift barrier is a theoretical cutoff point where basically the effective natural selection is negligible with by comparison to the effective genetic drift.
Okay.
When you're below that point.
Okay, thank you. So under that logic, then, if I were to give you 3 simulations, all of which are either truly neutral or effectively neutral, and the effectively neutral ones only differ by their effect magnitude, by their effect direction. So one is slightly beneficial, one is slightly deleterious, and then the third one is truly neutral. When you look at these 3 distributions, under your idea, these 3 distributions should look exactly the same, right? Do they look the same to you?
So if I'm understanding, you're saying the first one is not neutral in any sense. The second one is effectively neutral, and the third one is strictly neutral. Is that
Nope,
correct?
I'm saying there are two simulations here that are effectively neutral, one that is slightly deleterious, one that's slightly beneficial, but both effectively neutral. And then the third one is truly neutral. Can you tell by looking at these allele frequency trajectories which is which?
I mean, yeah, you're saying that the effectively neutral ones aren't behaving identically to a strictly neutral mutation. Is that your point?
That's my point, yes.
Yeah, I can definitely
so in that
grant
sense,
that.
it's not that drift is not overpowering selection. Selection is still adding a bias here, right?
Well, it's certainly overpowering. That's the difference. That's why
Is
it's
it?
effectively
So
neutral.
can you look at which of these and tell me which one is slightly beneficial? Can you tell which one it is?
Yes. So again, the point you're trying to make here is that you're saying effectively neutral mutations aren't identical in their effect with a strictly neutral mutation. So you're saying there is some amount that selection acts on it. It's not literally a zero amount. Is that correct?
Yeah, so selection is in fact acting and differentiating these mutations,
Right.
even though they're effectively neutral, right?
It's acting, but not in a way that is going to overpower the effect of drift. That's the whole reason we have a difference.
So can you tell me which of these, again, are the slightly beneficial one? Just A, B, or C.
So what are you showing me on the y-axis there? Is
That's
that
the frequency? Sorry. So these are 100 different alleles, and these are their allele frequency trajectories across 400 generations.
So I would expect that the beneficial ones would increase in frequency, the deleterious would decrease. So I'm going to say A would be beneficial and B would be deleterious.
So
B
is strictly neutral. C is the one that's deleterious.
Okay.
So notice how only one mutation in C increased in frequency. So based on these graphs, does it seem fair to say that you need proportionately fewer beneficial, effectively neutral mutations to compensate for slightly deleterious ones? Would you need fewer of them based on these graphs?
Sure.
Okay. What time are we at, Donnie?
You just hit the 3-minute and 30-second mark. So you got about a minute and a half.
Okay, so under your argument then, if we need much fewer beneficial mutations to compensate for the deleterious ones, is it true that having a preponderance of deleterious mutations necessarily means that you're going to lose fitness if you need much fewer beneficial ones to compensate?
No, it doesn't necessarily mean that. And I think you proved that with your sim, because your sim had 100 times more deleterious than beneficial, and yet it grew without limit. So yeah, it definitely does not necessarily mean that. It depends on your parameters.
Sure. So under these parameters, under these effectively neutral parameters, it seems like you would need fewer effectively neutral beneficials to compensate for deleterious.
Sure.
Okay, cool. So then I guess I'll take just a minute and ask you about some of your SLIM code. So unfortunately, I obviously didn't have a chance to look at this beforehand, but it would be nice to know the DFE that you used in SLIM. So
Absolutely.
How did you parameterize fitness in SLIM? Did you use a non-Wright-Fisher model? Did you use a Wright-Fisher model? How did you parameterize it?
So it was technically speaking, a non-Wright-Fisher model, I believe. However, I think the assumptions of Wright-Fisher wound up being created basically in SLIM. When you choose Wright-Fisher, my understanding basically is that it's going to automatically create certain assumptions. You can tool those yourself if you choose non-Wright-Fisher, if you select the right settings. And I think basically mine is still Wright-Fisher, except that the population size isn't stable. It decreases, obviously. My DFE is coming from Rossimo and Schreiber 2014. Let me see if I can bring that up here. I actually had the I'm sorry. I'll get that.
It's fine. Like if we're out of time, it's fine.
Yeah, it's Rossimo and Schreiber 2014, and that is where I got my DFE for the deleterious. It's and I'll be happy to show that here in the debate where I got that from.
Yeah, that's fine.
Gentlemen, good fast-paced cross exam. Of course, if there's anything to respond to there that you guys would like to, we still have rebuttals and Dr. Zach B. Hancock's negative constructive plus first negative rebuttal of 18 minutes. To the audience, anybody just joining us, I see people have questions. Yes, Dr. Zach B. Hancock has an opening statement. The way that this format is constructed is that we have Paul's first opening, then we jump right into a 5 to 6 minute cross exam like we just did. Now it's Dr. Zach B. Hancock's turn to give his opening plus first rebuttal. Then we jump into another cross exam. So, okay. Zach, whenever you're ready, just let me know. I do see your slides at the bottom. Would you like me to get those up?
Yep.
Okay. I've got them up on screen. I'll have you as the main presenter on the screen. If there's any issues, I'll let you know. So, okay, you got 18 minutes. The floor is yours. Go ahead.
Okay, so are mutational effects a problem for evolution? And I kind of want to begin with actually a quote from Paul and Dr. Rob Carter from an article they wrote in 2019 about information. They write, quote, "Darwinism requires that random mutations over long periods of time create decidedly non-random sets of information." And I think this is a really good framing because it allows us to directly compare the predictions of genetic entropy with that to, as Paul calls, Darwinism. So classically, when we think about Darwinism, what we mean is that random mutations are happening all the time and in all directions. However, there's a sieve that natural selection sort of sorts through that variation, and what you get out is a decidedly non-random trait. Something like the beak shape of the medium ground finch, right, is very much not a random trait. It fits its environment exceptionally well. So that's the prediction of Darwinism, that natural selection comes in and biases the randomness towards a particular direction. Now, genetic entropy here cries foul. They says, no, mutations are actually far too slight in their effects, and they actually will circumnavigate natural selection, and they will build up in populations and eventually degrade what selection or what a creator made in the first place. So you end up getting a beak with variance that just explodes. It's in all different directions, right? So in effect, what genetic entropy is about is the removing of the sieve of natural selection and assuming the supremacy of randomness, because it's basically claiming that drift and mutation are going to overpower the process of selection. And so from this toy example, we can actually formalize this and use this to help us predict what we would expect to see under these two different models. So genetic entropy, whether it's a trait, whether it's a genotype, allele frequencies, no matter what it is, basically supposes that that trait should change randomly over time because mutations are random and drift is random. And so you can model that as a kind of Brownian motion process where the variance of that trait just explodes across time. Now, if you plot this as a histogram, the average actually doesn't change, but you do see an explosion of the variance. So this is the base prediction of genetic entropy, degradation and randomness through time. Now, selection has a decidedly different prediction, that it should actually be shifting the mean trait in a direction towards something, whether that's adaptiveness or whatever, and it decreases the variance around that trait by basically purging individuals that fall in the extremes of those distributions. So you can imagine plotting these two distributions against each other, where the black is the expectation under just randomness, under genetic entropy, and then the blue is the distribution under selection. And notice how selection has, in effect, created something you would not expect to see under a purely random distribution. And so, in effect, the difference in the means of those distribution is the amount of information that selection has managed to extract from that random process. So to give you a kind of analogy here, imagine you had 26 letters, all the letters in the English alphabet, and you picked them at random, right? And also some symbols like semicolons, commas, etc. What you're going to end up with is a distribution that looks like this, right? Just a bunch of letters. Now compare that to the distribution of letters on the right. Now, that looks nothing like what you would expect to see under a purely random process. Now, importantly, it doesn't actually matter if you've read Shakespeare's Hamlet or if you can even speak English. The point is not the meanings of the words. The point is that the sequential arrangement of these words look nothing like the words you would get from a random distribution. Similarly, the shape of the beaks of a finch's beak looks nothing like a beak you would get under a purely random process. So it's the measure of the unlikeliness of what you see relative to randomness. So with that sort of toy model and sort of framing, we need to ask then the fundamental question, can selection do this? Can it add information even when mutational effects are extremely weak? And to do this, what we need is a model. We need a model of selection. That's how population genetics works. We need this model to be biologically realistic. It needs to include extremely weak mutational effects, and it needs to ultimately predict that information is going to be gained in this system. And we also need to be sure that it shows that it can work on biological systems as well, that it makes actual predictions. So let's set up the model. So all models have assumptions. And the first assumption of our model is based on the relationship between the genotype and the phenotype. How do we think those things are connected? So our model is not going to assume some one simple gene leads to one trait, as most of the models that Paul actually presented make that assumption. Instead, we are going to assume that complex traits actually have an omnigenic architecture, that they are underlaid by, in effect, every gene that you have that is at any point in time expressed in your tissues is contributing to the variance in this trait. Now, this is in effect an empirical observation. These are two recent papers that are pushing the field to move beyond basic Mendelian genetics and respect the idea that traits actually are composed of many genes all interacting with each other. So our model begins with that assumption. Now, from all the rest of the assumptions are basically corollaries to that one. If that one's true, these other ones must also be true. So allelic effects are on average extremely small because there's a ton of genes interacting, right? So each individual's effect must be very small. So down here, this is a plot of human height. These are all the mutations contributing to variance in human height. At 0, this is no effect. And then as what you can see is all of those variances are punctuated around 0, right? So very, very tiny effects. Over here on the left is corn oil production. So these are all the alleles that are contributing to variance in corn oil. And you can see there's a ton of them, and most of them fall under this line. So this line here is the line of statistical significance. If you fall below that, then you are very, very weak, right? You're not even rising to statistical significance. So lots of alleles contributing it. So again, this assumption is basically an empirical observation. Now, because of this, you've got a bunch of genes, all with tiny effects. That must mean that drift is going to dominate at the level of the nucleotide, right? That all of these are going to basically fall into the box of effective neutrality. Because again, they're tiny little bitty effects. Selection is not going to be very efficient on them. Now, the last assumption of the model is perhaps the most important that we understand. So selection does not see those individual mutations, right? It fundamentally doesn't. All that selection can see are phenotypes. It sees the ultimate expression of all of those individual mutations. I'm surprised Paul didn't bring this up, but Sanford likes to use the analogy of the princess and the pea, right? Where selection basically can't see the pea under all of those matrices. So that's what we're sort of thinking about here. But I want to sort of translate this and talk about the implications of what this really means. So what this means is that all alleles are dependent upon the genomic context in which they emerge. So for example, this is a little toy model where this curve is a fitness curve. Here in the middle are phenotypes with 0, and that's the highest fitness. So long as you have the phenotype trait of 0, you have the highest fitness. As you go away from that, you lose fitness. All these little things that you're seeing here are example haplotypes. And now you can have one of two different kinds of mutations. You can either increase the trait or decrease the trait. So now notice here are 5 different haplotypes that all have novel mutations, and yet they all maintain the optimum trait of 0. And this is because so long as you have an equal number of increase and decrease, they cancel out and you have the optimum trait. But notice that if you take those exact same mutations and you put them on a different genetic background, suddenly you've decreased fitness. You move yourself away from the optimum trait. So the key point here is that alleles don't have fitness effects on their own. Selection doesn't see them. Selection only sees their net effect. How do they all act together? That's what selection is going to pick through. And the last thing here is notice that all of these haplotypes have the exact same number of mutations. So again, it's not the number of mutations, it's their arrangement relative to their effects. So that's a really key idea that we need to be sure we understand. Okay, so now let's formalize the model. So this model was first derived by R.A. Fisher in 1918. It's over 100 years old. It's easily the most powerful and useful and widely applicable model in all of biology. Basically, everything you eat, you have this model to thank. He started off by deriving it based on the variance in the phenotype. So we'll start there. So thinking about phenotypic evolution first, you can imagine taking the phenotypic variance and he imagined decomposing it into a series of variances. Now, what we really care about is the genetic variance, right? The variance is being inherited across generations. So that's V sub G. We then break up V sub G into a series of component parts, the additive, dominant, and epistatic variances. Now, if you evaluate the covariance between parents and offspring using linear least squares, what you find is that the correlation between them, which is ultimately what selection is going to act upon, is simply the additive genetic variance. And this is why we classically define heritability as the additive variance divided by the phenotypic variance. And this yields the breeder's equation. So the average change in the trait from one generation is simply the product of the additive variance and the beta regression coefficient, which is the selective gradient. And you'll notice that this term is actually mathematically equivalent to the covariance term from the Price equation, right? Which Dan and I wrote about extensively in the paper. Now, since the genetic variance is mostly being shaped by drift, right, because the alleles are effectively neutral, then the net change that selection can enact in this situation is 2n, that's the bound to the population size, multiplied by V sub A times beta, right? So that's the net change that you expect. So how well does this fit the data? Well, up here on the top right, this is the response to selection in the first 50 generations. And then this is the effective size on the x-axis. This sigmoidal curve here, this is the expectation under the model. And then here's a bunch of empirical data. And what you can see is the shape of those empirical data fit it pretty well. Now, there's a bunch of different studies, but they follow the general shape of the model. They fall a little bit below it because these effective sizes are pretty small. These are like small laboratory populations, agricultural settings, that sort of thing. So there's a lot of linkage that kind of pulls it below. But for biological data, that's a pretty good fit, right? So the key takeaway here is that selection, despite all of the underlying alleles being effectively neutral, actually is extremely efficient at the phenotypic level. It's capable of driving phenotypic evolution, and we've seen that using the breeder's equation in livestock breeding for a century now. So this seems to suggest a paradox. Selection is capable of working at the phenotype level, but what's happening at the underlying genetic level? So a man named Alan Robertson in the 1960s resolved this conundrum by recognizing that you could take the average change or the net expected change of the phenotype, and then you could translate that into the expected change of the genotype via the probability of fixation. So the probability of fixation is 2s over 1 minus e raised to 4ns, negative 4ns. And so for a slightly beneficial, though technically speaking, effectively neutral mutation, so n of s is equal to 1, then the probability of fixation of that single mutation is 0.002. That's really tiny, right? That's a really, really small probability of fixation. But if you think about it relative to neutrality, it's actually 4 times more likely to fix than a purely strictly neutral mutation. And for a slightly deleterious mutation of the same effect size, so n of s is equal to -1, it's 54 times more likely to fix. And so if you look here, this is the full range of effective neutrality from 1 all the way to 0. And this is the ratio of selection beneficial to negative. And you can see across this entire distribution, you are far more likely to fix an effectively beneficial. And so what selection is in effect doing, and this is why I kind of talked Paul through the distributions that I showed you in the cross exam, is that what selection is doing at this level is it's slightly biasing the probability of distributions. It's biasing it such that it removes the harmful alleles and favors the beneficial. So down here, this is the expectations under neutrality, and compare that with under selection. And again, despite all the noisiness, it's able to generate a distribution that's biased away from neutrality. And this is also true despite the preponderance of deleterious mutations. Now, Paul made the argument that the simulations that we used were outside of the expectations under neutrality. But what's really curious about that to me is you can see all of these alleles, right? If you basically cut off to 0.01 or 0.001, all of these alleles will fall along that level of neutrality. And definitely all of the beneficial ones fall in that level of neutrality. It's also important to note that the majority of distributions of fitness effects are not just nearly neutral. There's lots of deleterious mutations in true distribution of fitness effects. And I also should point out, we didn't just use one study. We used 4 different studies. It's just one of the other studies we cited cites 4 distributions in it. So That's technically not true. And despite the fact that the wild preponderance, we used 99% deleterious mutations, if you look at the ones that actually go to fixation are mostly beneficial, right? So they're basically overwhelming the positive ones, or the damaging ones. Now, again, just in case you don't like our simulations, let's go back to the person that originally founded nearly neutral theory, Tomoka Oda, and see what she says about it. So these are her simulations from the 90s. These little red bars are the ones that are deleterious that fixed, and then all the rest in her simulations were fixed beneficials, despite the fact that she uses the effectively neutral distributions that Kimura first derived by just including a slightly advantageous side. So she uses the distributions and manages to find the same exact thing we did. So don't take my word for it. Take the originator of nearly neutral theory. Okay, so let's bring all this back together. So just in case you think this is weightless elephants on frictionless surfaces, over here on the left, we've got the GWAS of corn oil production. So you've got tons and tons and tons of alleles, all with very, very small effect. And yet, on the right is the long-term selection experiment showing the response to selection. So here in the dashed line, this is the control line where there's no selection being added at all. So this is just drift. And then here in the dark green is what's actually happening via selection. So you see a consistent and persistent response to the selective pressure. And indeed, what this difference represents is that distinction between what is expected under randomness, that is to say under genetic entropy, if all the alleles are effectively neutral, versus what we expect to see when selection actually is able to sort through that variation. Right? Now, again, just in case you think agricultural setting, that doesn't count, here's a study of Darwin's finches. This is the model that they used. This is the exact same phenotype model I showed you. And they were looking at the change in beak shape across drought years. So this is the observed versus the predicted. You can see an incredible fit to that. You very rarely see this good of a fit in biological data. Down here, this is for several different traits that they measured. There's the mean predicted versus the mean observed. You can see it matches it exceptionally well. So the model also works well in natural populations. And this is just the GWAS to show you that beak shape as well as beak size are determined by a ton of alleles spread out all across the genome. So somehow selection is able to sift through those. Now, the last thing that I want to argue is that there was a theoretical investigation of the infinitesimal model, and they were basically asking, when is selection the most efficient at driving adaptation? Is it when it's very, very strong, or is it when it's extremely weak, like in the case of the infinitesimal? And what they found was that the infinitesimal model, as being able to divorce sort of randomness from information from noise, they write, while selection cannot purge weakly deleterious mutations under the infinitesimal, it can perturb the allele frequency distribution of each by a small amount and thus shift the distribution of higher level traits, so beak shape, corn oil production, those sorts of things, very far from neutrality. In fact, information accumulation and maintenance are most cost efficient in this regime. So this is the measure of effective neutrality down here, the amount of accumulated information, and you can see it is maximized whenever selection is very weak. So just to give a quick closing summary, I've provided us with an objective quantitative way of comparing the genetic entropy hypothesis, which is just based on randomness, with that of Darwinism, which should extract information from it. I then gave you a quantitative genetic model that assumes alleles are nearly neutral, demonstrated its applicability by applying it to empirical studies, and then I gave you a final theoretical study that shows that it's the most efficient way of accumulating information. So effectively neutral mutations, even when they're bombarding populations, are thus no problem for evolution. And I think that's my time. Thank you.
Dr. Zach Hancock, thank you very much for that negative constructive plus first negative rebuttal. 18 minutes really does fly by. We've concluded our affirmative constructive of 15 minutes, cross-exam of 5. This session here of 18 minutes, we're now moving into another round of fast-paced cross-examination. We have another 5 minutes. This time, Paul, you get to lead the way. And just make sure to unmute yourself, and I'll start the timer on your first question. 5 minutes. Go ahead.
All right. Dr. Hancock, you stated that the DFE produced by Basner and Sanford made it obvious that fitness would decline. You didn't even need to do the simulations to see that. What was it? Can you be specific? What was it about the DFE that they used that exactly would cause that effect? What caused you to say that?
Yeah, so I said that because the proportion of beneficial mutations they used was 0.001, which is smaller than any empirical distribution that I've seen.
Okay, so that's 1 in 1,000 then, is that right?
Mm-hmm.
Yeah. Okay. So the reason why the DFE that they used would result in fitness decline just came down to the ratio of deleterious to beneficial then.
Correct.
Okay. Do you believe that you have solved Kondrashov's paradox?
Me personally?
You
No.
personally, yeah.
No, I don't. But it has been solved, and I'm happy to talk you through it if you'd like.
But the simulation that you provided did not solve it?
No, and it was not even set to try to solve it. That was not,
Okay.
the simulation was trying to solve or trying to answer the Sanford paper. That's why it was set up in the way that it was. We were trying to mimic what Sanford did, not what Kondrashov did. That's a whole nother conversation.
Okay. Can you provide us with any citations at all to support your use of the figure of 7 billion for the human effective population size?
Yeah, so that's just based on our population size, our current population size. The argument that I was using is long-term effective sizes is what's called the inbreeding effective size or the equilibrium effective size. And that's the effective size that stabilizes through time, right, and reflects long-term events into the past. But selection happens today between parents and offspring, right? And so the effective size that matters to selection happening right now today is what's called the variance effective size.
Right, but what is your citation that would back up your claim of the figure 7 billion for effective population size?
Oh, I just told you. It's because I'm basing it on the variance effective
Right.
size.
You told me
which is
what you're
close to the
doing.
census size.
Yeah, you told me what you're doing. You did not give me a citation. Do you have one?
It's the population size. That's my citation. How many individuals there are.
All right.
Google it.
do you-
Like we know how many
okay.
people are on the planet.
Do you consider your published simulation to be a oh, sorry, I already asked you that one. Let me see if I have any others, or have I already asked them all? Oh, okay. When you created your DFE, were you aware that you were choosing parameters that minimize the presence of effectively neutral mutations almost to nothing? Or are you learning that for the first time tonight?
I do not know where you got that from.
Okay.
That makes no sense to me, especially given the shape of the distribution. It's completely stacked around zero. So I have no idea where you got that from.
Okay. Well, I'll explain it again. I explained it in my initial 15-minute, but I'll take some more time on that. Is there anything else? Do you agree or disagree with Dr. Lynch that any claims about evolution must be held in doubt until they can be shown to be theoretically feasible?
Yes,
You agree?
of course.
Okay. Donnie, where am I at on time? I think I've exhausted my questions here.
Well, you just hit the 3-minute and 40-second mark. So technically you got to.
Okay, let me ask another bonus question. This isn't directly relevant, but do you agree with Dr. Lewontin, who stated that scientists must assume or use only purely materialistic theories to avoid allowing a divine foot in the door? His words, not mine. Would you agree that it's absolutely unacceptable to ever infer any supernatural causes in science?
Can I infer I don't know how one would infer a supernatural cause because I don't know how to test a supernatural cause. It seems like especially if that supernatural cause is all powerful by definition, it can do anything. And so I could invoke it to explain anything. And that to me degrades the value of science.
So okay, you would agree that it's inherently materialistic in nature, science is. Okay.
Yes.
That was a bonus question. Donnie, I think
That's
I've
good though.
done
No, that's 5 minutes. So very good, gentlemen. Excellent technical debate so far. I think this is a very valuable debate. I know it is to me because I do like to dig deep on these technical topics and what an important topic this is to the audience. Thank you again for the interaction and engagement. I'm all caught up on your questions and a few of the challenges or objections that have come in as well. We are looking to make this debate as comprehensive as possible. So at this point, I think we've done a good job at setting the foundation. There's a lot of points on the table, a lot of arguments put forth. So if you disagree with those points in the live chat, whether you're on Paul's side or Dr. Hancock's side, send those in the form of questions. We look forward to engaging them in the Q&A. Okay, our next session here is the first affirmative rebuttal. 8 minutes. Paul, that would be you. So feel free to let me know when you're ready. And
all right, there's so much to cover and so little time. Let me see here, because this time I'm going to share my screen rather than going in the other, like that other way that I did it before.
Okay.
So
I won't start your timer yet. I'm just
gonna share my entire screen so I don't have to fool around with trying to select a specific tab and all that. Okay, so let us go now to. Oh wait, am I sharing?
Well, I see it at the bottom, but I'll wait till your slides get up.
Okay, okay. So if you can see my screen now, so I'm just gonna go ahead. If you'll put the sharing up there.
Yep, it's up there, and then it's up to you if you want to go full screen, just to see the text.
I'll go full screen here.
You could also click hide on the bar too if you'd like.
And just don't forget to give me that 1 minute reminder, because I'm going to have a lot to try to cover in a short time here.
Okay, no worries.
Thank you. I'm ready to go. Let me know when to start.
The floor is yours, starting now. Go ahead.
Okay, so real quick, what we just discussed in the cross exam. I'm going to answer Dr. Hancock's question of where am I getting the percent below the drift barrier for his DFE. Now that comes from the parameters that he used in his equation, which is a gamma distribution. What you're seeing on the screen here is a gamma distribution calculator. You can use these for free online. You can Google it. But basically what you're going to need is your shape and your scale parameter. Now shape is, I believe, for Dr. Hancock's DFE, he chose actually 0.5 for the shape. And he also chose 0.5 for the average effect size. Now you can see up here in the upper right, the scale is actually a function of the average effect size divided by the shape parameter. So when you do that for his DFE, you get 0.5 for the shape and 1 for the scale, because he used the same number for the shape and for the average mutational effect. Now, the drift barrier, I'm using Kimura's drift barrier 1/2n. So that's 0.0005 for this simulation. That means that for his DFE, we're looking at 2.5% effectively neutral, almost everything selectable. So that's where this blunder has come from. This is nothing at all like, and these parameters are nothing at all like what you will find in the literature when you're looking for these specific parameters. Now I'm going to move to some more slides here. So let's see, view. Okay, here to answer Dr. Hancock's question about my parameters, because I did show his parameters compared to empirical human. Now I'm showing my parameters compared to empirical human. And unlike my opponent, I did apply scaling logic. So that means I divided and I multiplied to create an equilibrium of sort of evolutionary dynamics, so to speak. And to get this to run, I did have to reduce the genome size by 1/3 as well, bringing me down to 1 billion base pairs instead of 3 billion. And that means you have to multiply the mutation rate by the same amount to get the same number of mutations per individual. And that's what is going to give us our load. And so here is my parameters by comparison. Now here is the DFE that I'm appealing to, which is this is highly important. This is a genome-wide DFE. And this was published by Rossimo and Schreiber 2014. And this is giving us an estimate of the human DFE genome-wide. That's what you see in the black dot or dashed line there. And then you see the other subsets of the genome. The most important other subset would be the red one, which is non-synonymous mutations. Those are the actually less than 1% of the genome that actually affect a protein a protein outcome in the code. So those are going to be the most powerful mutations of the whole lot. And in one of the papers that my opponent cited in his work, which is Walker, let's see, it was Walker et al. 2006, I believe. They actually give us their parameters for that 1%, and they have a mean effect size of 4.3% or 0.043, and their shape is 0.23. So even for the 1% most effective mutations, or I'm sorry, most powerful class of mutations, it's far, far less than what my opponent used. I'm going to skip through some of these. I actually already made that point. Now, at a realistic human affected population size of 10,000, Hancock and Stern Cardinal's DFE contains only about 0.8% of effectively neutral mutations, which is insanely low, essentially none. Compare that to Walker and Kitely. If I apply their more effective DFE, that's going to be 16.6%. But however, using the DFE from Rossimo and Schreiber, which again is genome-wide, what you can see is that the mean effect is actually extremely low, which actually agrees with something my opponent stated. I was surprised to hear him agree that nearly all mutations are effectively neutral. Well, if that's the case, why didn't you model that? That's odd. Most new mutations are neutral with respect to survival and reproduction, and therefore are irrelevant in terms of natural selection, but not, it must be pointed out, to evolution more broadly. Well, it turns out my opponent is actually promoting something that agrees with this. Widely accepted that most new mutations are neutral. Okay, my opponent is granting one of the most important things in this debate, which is the effective neutrality of the distribution. Unfortunately, he did not model that with his simulation at all. And when he says that he has not solved Kondrashov's paradox, I can only say amen to that. He absolutely has not solved it. Where he is incorrect is to try to state that it has nothing to do with genetic entropy. In reality, if you read the book Genetic Entropy, Kondrashov is all throughout it, and it's very clear that the mechanics that Dr. Sanford are appealing to are in fact Kondrashov's mechanics. Now, it is here. This is important too, because we're talking about the ratio. Is it 100-to-1 deleterious to beneficial, as my opponent used, or is it actually 1,000-fold? Well, the citation that he used in his paper, San Juan et al. 2004, actually say it's 1,000-fold. They state this: it is generally accepted that beneficial effects are 1,000-fold less common than neutral and deleterious ones. Therefore, it is striking that 2 of 48 random mutations were beneficial. However, this result is not so surprising, so they're not trying to overturn the consensus here. If we recall that we used a chimera genome as a template for our mutagenesis experiments. So this is kind of a special case, and certainly is not an attempt by this author or these authors to overturn the consensus of 1,000-to-1. It's interesting to me that he cited
1
this
minute left,
but didn't use the ratio that they argued for and said was a consensus. I'm also going to say that I ran the simulations to test for epistasis, since that was Kondrashov's attempt to explain this away. It turns out that empirically, when you run the sims, epistasis actually speeds up the decline. So sorry, that one doesn't work either. And thank you, Donnie. I'm going to close at that point here.
okay. Paul Price, thank you very much for that 8-minute rebuttal. We're now moving into our rebuttal from Dr. Zach B. Hancock, taking the negative for tonight. So, Zach, whenever you're ready, I do see your slides at the bottom again. Did you want me to pull those up for? Okay. All right. You have 10 minutes So the floor is yours. Just make sure to unmute.
All right,
Go
sorry,
ahead.
just getting my timer set up here.
No worries. Take your time. To the audience, keep those questions and challenges coming in as we're moving very close to the Q&A. We have this rebuttal here of 10. Then we wrap it up with a second affirmative rebuttal of 5. And then we're going to hang out with you guys in the audience for a bit. So, Zach, floor is yours. 10 minutes. Go ahead.
Okay, thanks. So it's very curious to me. Paul has already admitted that selection works on effectively neutral mutations. We kind of began that. So whether or not Dan and I modeled them correctly is actually irrelevant to the conversation because selection operates on effectively neutral mutations, and Paul's already admitted as such. And actually, you don't even have to look at our paper. You could go all the way back to Tomoka Oda, who modeled the distribution that Kimura based it off of. So it's not like we are the first people to do this. So there's really no reason that we're even spending so much time talking about our distribution. But I'm perfectly fine saying that the distribution doesn't reflect it. I don't think that's true. I would have to go back to the actual SIMs, but I don't think it's true. But I'll grant it. But it actually is completely irrelevant because the model that I presented assumes every mutation is effectively neutral. That's the model I gave Paul in this debate today. I didn't present my model. I presented the infinitesimal model that Paul has not said a single word about. He has pretended that I have not even mentioned it at all. So I'm going to go back to his opening, talk through a few of those things, but I want you to remember in my presentation, I gave you a model that fits the data, that has been used to affect every single thing that you eat. Your entire lives have been shaped by it, and you have enjoyed modern agriculture from it. And Paul is saying it doesn't work. That's his argument. So let's now talk through his opening. So he showed Kondrashov. He really likes the Kondrashov model. Let's talk about the assumptions of the Kondrashov model. Kondrashov basically assumes that one gene equals one trait, that each gene has a fitness effect that you can measure that impacts one single trait. What the real data is showing, what the omnigenic model is showing, what most modern genomics and developmental biology is showing, is that that's not the case, that traits are highly polygenic, impacted by many, many different alleles all across the genome. And this is actually the response to Kondrashov that Brian Charlesworth wrote in 2013. In his paper, which is cleverly called Why We Are Not Dead 100 times over, he just states why we aren't. And it's because if most traits are actually polygenic and not simple, because Paul seems to think that traits are very simple and it's like one gene, one trait, that doesn't seem to be the case. If traits aren't simple, then actually the genetic load collapses to what's called the genetic variance. So on given stabilizing selection basically acts to remove the excess individuals that fall along the end, and it stabilizes it around a mean. And so the genetic variance is basically an equal measure of the load. When that is the case, then the mutation load paradox or the stochastic mutation load paradox vanishes because the genetic variance among basically all life is really tiny. So the load is really tiny. Another key assumption of Kondrashov's model is that it assumes viability selection after individuals have extracted resources from the environment. So if you basically enable selection to act at the zygotic level, or to act at the gametic level, or to act before that individual has impacted the rest of the population, the problem falls apart. It doesn't exist anymore and you resolve it. Another key problem is that the stochastic mutation load suffers from the same problem as Haldane's original load, and that is it bases the load on a hypothetical individual that doesn't exist. And this is the thing that's kind of frustrating, that Paul showed nothing but quotes and he didn't show the stochastic load model math, is that the way the math works out is you imagine an individual that has no mutations at all, none, no effectively neutral mutations, and you subtract from that the average of the population and all their mutations. And then that difference is the load. So basically, you're comparing the fitness from a Superman that has no mutations and the average population. That is not actually how fitness is evaluated. Individuals in real populations are not competing with some idealized Superman. They are competing against equally loaded individuals. There's tons of studies that show there's actually no clear correlation between the extent of the genetic load of a population and whether or not that population will persist through time. Because again, these ideas are decoupled from each other. What we actually should be measuring is the actual load of individuals in the population relative to one another, not to some hypothetical individual. And that has been the critique of Kondrashov's paper from the beginning. And in fact, if we want to cite people, I will cite people way more famous than Kondrashov. Take Sewell Wright, take Theodosius Dobzhansky, who have critiqued the genetic load concept from the very beginning based on the oversimplification of the relationship between genotype and phenotype. It assumes it's one-to-one, that you have an allele that has a fitness effect that then affects that phenotype. And it assumes that you're comparing supermen, non-mutated individuals with the average normal mutated individual. The fact of the matter is there's been no one in the population that has not been mutated, right? Everyone has a genetic load, and they always have. And so it's just not even a realistic comparison. Furthermore, we talked a little bit about the no selection zone. Obviously, there is actually no such thing as a no selection zone. Effectively neutral mutations, if they are slightly beneficial, always fix with a higher probability than slightly deleterious ones and can compensate for each other. Again, you don't have to take our paper's word. Take the papers from the 1990s and the 1980s, go all the way back to Kimura 79 if you don't believe us, and you can go on their distributions. Pick your favorite distribution. The fact of the matter is that selection can and does act on effectively neutral mutations. Another thing is that he basically goes back and forth between neutral theory and nearly neutral theory. He cites Lynch several times as talking about that the beneficial must equal, or by talking about that the rate of fixation is basically equal. That's only under strict neutrality is the population size irrelevant. The population size is not irrelevant under near neutrality. That's the whole idea. But he uses those things interchangeably several times. What else? So the big questions that I have are left are basically around the parameterization of his model. So he says that he used a Wright-Fisher model that he changed to a non-Wright-Fisher model, which makes me wonder how you actually parameterize selection itself. So a Wright-Fisher model uses selection that's based on, it's a kind of soft selection actually, so individuals can have tons and tons and tons of offspring, right? Whereas in a non-Wright-Fisher model, it's absolute fitness. It's based on viability. And so actually our model is a little bit closer to the classic genetic load model than a model where you've basically tweaked a Wright-Fisher. So you'll have to clarify exactly how that worked, how you pulled that off. Let's see what else. So again, what we were trying to model there was Sanford's model. We were trying to get as close to Sanford's model as possible. And the reason that you see the perpetual fitness increase is actually like, we talk about that in the paper. It's called a Darwinian demon. And I reference it in the paper that whenever you have a constant supply of beneficial mutations, then the population basically will just explode in size, right? The way actual populations work, again, based on the infinitesimal that I had talked us through, is that you will climb a peak. And once you reach that peak of fitness, any additional change pulls you away from that peak of fitness. You don't just have continuous beneficial mutations that just pile up forever. That's not really what you expect to see. And I can sort of demonstrate that, or I guess in my last couple of minutes, Donnie, if you'll share my screen, I want to just walk through a couple of final quick things. Okay, yes, so some failed predictions of genetic entropy that I want to be sure that I get in here. So under genetic entropy, we would expect that populations harboring the most genetic diversity should show the greatest decline in fitness, right? If you've got a lot of genetic diversity, then you should have really low fitness because you got a lot of mutations. The exact opposite has been seen. We do not see that at all. Furthermore, large-bodied vertebrates should be incapable of tracking fitness peaks. Because again, selection should be so weak, they should be overwhelmed by mutations, they shouldn't be able to track fitness peaks, and yet they can. We have study after study where individuals are being able to track fitness peaks in adaptive landscapes. The third thing is that selection cannot act on traits determined by effectively neutral alleles. That's what I sort of talked through and demonstrated that's not the case. And again, under the infinitesimal model, that is exactly the way selection works, and it is when selection is the most efficient. And then finally, there is no clear connection between the genetic load and population persistence. And this is true whether it's Kondrashov's model, whether it's Haldane, go all the way back to 37 and Haldane. The reason that this is called a paradox is because their math seems to show it, but reality doesn't. We don't see this happening in nature. We do not see perpetual fitness decline in populations with high loads. And we've directly compared the genetic loads between populations, and we do not see differences in persistence. And this is because, again, the underlying assumptions of the load model are far more complex than what Paul is presenting here. You have to make assumptions about when is selection acting? How is it acting? Is it soft? Is it hard? Is it acting on zygotes? Is it acting on individuals? Is it acting on individuals before they get resources, right? Or are you using the classic load model like Kondrashov and Haldane, where you're comparing a Superman to the average of the population, right? All of these assumptions you have to investigate because the paradox suggests that it's not actually happening. We don't see it in real populations. Okay, and I'm out of time. Thank you.
Dr. Zach Hancock, that flew by. Okay, let's bring Paul back in here. And that was 10 minutes. We're moving along smoothly. I'm all caught up on audience questions, challenges, and objections. I look forward to seeing how Paul and Zach answer these questions and interact with some of the challenges that have come in. Okay, Paul, you have now second affirmative rebuttal. So 5 minutes. I see that you're looking to get your screen shared so we can, would you like me to put those up right now?
Yes, please.
Okay. They are up and you're good to go for 5 minutes.
All right. Thank you, Donnie. And just to close here, my opponent has put out a few straw man arguments tonight. First of all, the claim that genetic entropy assumes that natural selection does not act at all and everything is completely random. That is, I don't know how you could possibly think that reading Dr. Sanford or reading Kondrashov. That's not the case. We're talking about a drift barrier, or even in SLIM, there is no literal drift barrier, but it's just a smooth gradient of less and less selection and more drift. And what we see in the results is the results of fitness decline. That's why we have this problem. The evolutionists managed to refute themselves decades ago without any help from the creationists. It's kind of ironic. The science media simply choose not to report on it, but I'm reporting here tonight because I want everyone to know evolution doesn't have a mechanism and it doesn't work, and they're just sitting on it. Tonight, we've seen that my opponent can by no means refute GE. He has made the following claims, which I have now refuted. 1, he's claimed that effective population size is 7 billion, cannot provide a citation. Nobody thinks that. It's 10,000. Check the literature. Mean selection coefficient, he claims, is 0.5. Again, far off the mark, off by 72,000 times what the literature says the genome-wide is for humans. It's way off. Shape parameter, 0.5, again off. It's 0.38 in Rossimo and Schreiber. It's 0.23 in Iyer Walker and Wolfit from 2006. It's just, again, not accurate. Deleterious to beneficial, he has it at 100 to 1. Wrong. It's 1,000 to 1 at the most, and that's from consensus view attested to in his own citation of San Juan et al. Theoretical problems can be hand-waved away? No. Dr. Lynch militated against that. It is simply not the case that Kondrashov's paradox requires genes to work simply on a single phenotypic trait 1-to-1. I don't know where Hancock is getting that. It has nothing to do with GE at all. GE only requires that most mutations are effectively neutral, which my opponent turns out to acknowledge, but did not simulate at all in his simulation. Under effective neutrality, the difference between the strictly neutral and effectively neutral in the population is negligible. It's not nothing, but it's negligible. This is exactly what we see in the SLIM results. I also want to thank, since I'm coming to the end, I'm thanking my friend Dr. Chris Roop, who I considered now to be the standard bearer going forward of Dr. Sanford's research and work on genetic entropy. I encourage people to check back to Genesis Out and Dr. Chris Roop. But with that, I think I've covered most of what I can in 5 minutes. I thank everyone for listening tonight. I thank everyone for being willing to go with us on this highly technical debate. And it was over in just a moment, obviously, but I think it's so important. So thank you again, Donnie, and looking forward to the Q&A. Thank you, everyone.
Paul Price, thank you very much for that 5-minute second affirmative rebuttal. So we've moved along smoothly. That debate flew by. I appreciate how well prepared the both of you were and how well studied clearly. Now, for those that enjoy, I'll give our debaters a 30-second break here before we engage your questions and challenges. For those that enjoy origins-related debates, we have another one coming up on Saturday. This is the great Grand Canyon debate between Jackson Rowe and Brian Lauer. Of course, tonight, excellent exchange between Paul Price and Dr. Zach B. Hancock. Last month, we had a fantastic marathon debate that went for 5 hours between Gutsick Gibbon and Dr. Peter Line. And more into the summer, we have this one that I'm pumped for: the proper perspective of geology, plate tectonics, or hydroplate. Dr. Howard Feldman and Brian Lauer. So we got lots to look forward to in the world of origins. Okay, gentlemen, let's get into some of these questions. So whoever the question's for, let's say it's for you, Paul, you get to respond first. Dr. Zach Hancock can offer his thoughts, provide a rebuttal if he'd like to, and then we hand it back to you, Paul, for a final word, and that way we can move along nicely. Okay, here we go. Let's start at the beginning. This one comes in from Jackson Rowe. Thank you very much, Jackson. Here's the question. I'm assuming it's for you, Paul, as Jackson's been rooting for Dr. Zach Hancock's side. So he's saying, at what point will genetic entropy cause extinction in a species? And if it won't do that, or you can't figure it out, why should we argue this?
Okay, the answer to that is if you watch my initial 15-minute presentation, I told you it maxed out at about 40,000 years for realistic human parameters, assuming 20 years per generation. However, I made the mistake of actually using too much of my opponent's code, because he actually applied fitness scaling in both directions. So his fitness scaling, and I use the same thing in mine, but his fitness scaling pushes down if you're over your carrying capacity. It pushes down on fitness, and it actually pushes up on fitness if you're below carrying capacity. To be realistic, that doesn't make sense. We shouldn't be pushing up on fitness just because you happen to be below carrying capacity. So, I would say my simulation could be improved upon by deactivating that density-dependent scaling on the upward side, and that would probably reduce the amount of time to extension by quite a bit, maybe as much as a factor of 2. I don't know. So maybe only 20,000 years max. It's certainly not compatible with deep time. And we absolutely can, thanks to my opponent, bringing the SLIM to my attention, and I had never used it before, but yeah, it's actually quite a powerful tool. I love it. And it actually allows us to estimate this exact thing. So I've been asked this before, and in the past, my answer was always, it's too complicated, I don't know. But now I can actually tell you, it's somewhere between 20 and 40,000 years for humans.
Thank you, Paul. Dr. Hancock, floor is yours. Go ahead.
Yeah, I'm curious about the idea about the density dependence. Presumably, like, the idea about fitness is how many offspring you have. And when you're in a non-Wright Fisher model and the environment is like, you're very far from how much the environment can hold, there's a lot of resources around you, then individuals should quickly grow in size to reach that resource optimum. And then once you've reached the resource optimum, that's where the mutations begin to drag you away from it. So I think that the density dependence is a realistic component of the model. But again, none of these are realistic models, right? To be very clear, what we were trying to do is model Sanford's as close as we could. But even his model is not real because there's no optimum. There's nothing you're trying to get to, and you're also assuming that individual mutations can be seen by selection. That is to say, you can apply a fitness effect to individual mutations, and that's what I meant by saying that it's a one-to-one. Because if a phenotype is determined by many genes, then that have different effect sizes, and actually, if you use SLIM, you can do this. Go in and look at QTLs and quantitative traits. Basically, what you will end up doing is you won't be applying selection on alleles. You'll be applying it on phenotypes, and you'll get different results.
Thank you, Zach. Paul, question was for you. Therefore, you get the last word. Go ahead.
Yeah, my opponent already acknowledged that most mutations don't have an effect on the phenotype. So I don't know how that overturns this problem at all. It just reinforces the problem. That is the problem, that most alleles do not have, considered individually, most alleles do not have an effect on phenotype. That's the problem. That's the source of Kondrashov's paradox, and thus the source of genetic entropy. So far from overturning it, this is actually, in a surprising twist of fate, confirming it.
Thank you, Paul. Okay, now we have one for Dr. Zach B. Hancock. Comes in from Geology Rocks 7. Appreciate the question. Question for Dr. Hancock. What was your reasoning for using a mean fitness effect of 0.5 to model the deleterious distribution of fitness effects in SLIM? What empirical studies can you cite?
So what we were basically trying, and it's important to notice that the negative 0.5 is the average effect of the gamma distribution on the left side, right? And what we were trying to do is include lots of very harmful mutations, because there are lots of lethal mutations. So you want it to actually capture that. We were also trying to match the shape of Sanford's graph. So Sanford has that graph that has that narrow thing. We were trying to match that shape. So it's trying to match the shape of Sanford's graph, as well as trying to account for the fact from like Adamir Walker or Keightley and Walker that there's a lot of actually pretty harmful mutations in humans. The important component is actually the beneficial side. The average effect was 0.001, right? So despite the fact that we modeled a very large deleterious distribution, the beneficial one, the average effect is actually really, really tiny. And there's only 1% of them, which is actually not a small percent given the various distributions of fitness effects that we cited in the paper.
Thank you, Zach. Paul, floor is yours. If you had anything you'd like to add, go ahead.
Yeah, I would just again state that, number one, the sources that he cited, I said in my initial, the only thing you could get was that San Juan et al. was about 50%, but that's not relevant to large multicellular eukaryotes, and it's a tiny sample size, and the rest of the DFE is all wrong. So if he was trying to match the shape of Sanford's DFE, well, Sanford was just using chimeras, and he stated that. And the parameters in Hancock's sim don't even remotely resemble chimeras, and they certainly don't remotely resemble the ones from the actual literature about what the genome-wide DFE in humans might look like. Not even in the same ballpark. So, yeah.
Thank you, Paul. Zach, you get the last word. Go ahead.
I mean, no, but again, we're trying to match Sanford's distribution, right? That's the distribution we're trying to match. And in fact, we were putting together various distributions. I gave an Arabidopsis paper as well. I don't know why you didn't cite that. I gave the Arabidopsis paper. I gave the San Juan paper. I gave a Keightley paper. I gave multiple papers and was trying to make a combined distribution of these effects. And again, you don't even have to base it on ours. If you don't like ours, go back to Tomoka Oda from the 1990s, go back to Dan Hartle from the same time, who showed the exact same thing using Kimura's distribution. So again, like the entire purpose of our paper was not even to show something novel. We concluded that paper literally by saying we didn't show anything novel. We have recapitulated what all the previous population geneticists have shown.
Thank you, Dr. Hancock. Okay, next question or challenge comes in from Dr. Dan from Croatia Myths. He's coming at you, Paul. Clearly, he disagrees with you here on genetic entropy. So I'd be curious how you respond. Croatia Myths says, another way to say it is that Paul is conflating neutral and nearly neutral, lumps them all together as unselectable, and he thinks it's silly. Paul, what are your thoughts?
I mean, I don't know how he can say this because I use Slim. So is Slim doing this? I don't think Slim does that. I don't think Slim actually even has any neutral mutations at all. In fact, it's nothing in the distribution has an effect of 0. It's a gamma distribution, and it approaches 0, but it's not ever there. So nothing that I put in my Slim code would be doing anything like this. My opponent has made much of this claim that selection still acts on effectively neutrals, but nobody disagrees with that. Dr. Lynch, who is a proponent of neutral theory, wouldn't disagree with that. He acknowledges that. It's just that the effect of selection is negligible and it is overwhelmed by the effect of drift. That is what the problem is stemming from. So, no, the claim is not that effectively neutral are identical to strictly neutral. That is not the claim. But the proof is in the pudding. If you think you can demonstrate this, then you need to go back to the drawing board and make an accurate simulation. That's what I did. And the result was genetic entropy.
Thank you, Paul. Dr. Zach Hancock, floor is yours. Go ahead.
I don't think he's talking about your simulations. I think he's talking about your quotes. So you did lots of quote mining. The most of this was quote mining. And when you quote mine, you're actually conflating many different statements. Some of those statements are about near neutrality, and some of them are about strict neutrality. And those are different theories that predict different things. That's what he's talking about there. The other thing that I'll say is that I actually did show that selection is capable of acting if every single allele is effectively neutral. That's literally what the infinitesimal model assumes. And it is the most powerful model in all of biology. If you don't like it or you don't think it works, then stop eating any agricultural product, or at least explain to me how it manages to both make all of these predictions and still operate on effectively neutral mutations. That was what my statement was about. That's my opening statement here, was about the infinitesimal model. And you have yet to mention it at all or even try to address it.
Thank you, Zach. Paul, it was directed at you, so you get the last word. And Dr. Dan here in the chat is saying that he's confirming what Zach is saying about the quotes. Go ahead.
Yeah, I will say it is a pet peeve of mine. Evolutionists constantly accuse creationists of quote mining. When you make that accusation, what you're essentially saying is that I took a quote out of context and I misrepresented the intended meaning of the author. So if you're making a claim like that, you've got to back it up. You can't just throw it out and say I was quote mining. And in fact, I would also answer the claim that the quotes that I had were, unfortunately, it's quite common in the literature for people to speak in a shorthand way and just say neutral. But if you read the literature closely, it is well understood that no mutations are strictly neutral. All the way back to the 70s, Kimura's 1979 paper had 0 strictly neutrals, and that is what is confirmed by the Eyerwalker and Kitely paper that my opponent cited in his published work. So no, when these scientists say neutral, they don't mean strictly neutral, they mean effectively neutral.
Thank you, gentlemen, for that engagement. Examining Origins, $20 super chat. Thank you very much, Rebecca, for the super chat support and question. I think it's for you, Zach. Don't polygenic traits make the evolution of genes even more difficult? We're straining gnats over slight benefits in already existing genes. But what is the evidence for mutations actually building all of this genetic information? Zach, go ahead.
So the way that I used information was about the distinction between random and non-random distributions. And that's basically how selection manages to extract information from random distributions. If we're interested in how new genes emerge, that's a completely different conversation. So how new genes emerge are by 2 processes, either de novo gene origin, we have lots of evidence of this, or it can emerge via duplications and neofunctionalization. And especially when we're talking about polygenic traits, a lot of this is actually by what's called subfunctionalization. And this is how all of those genes end up getting linked together. To give you an example, between all 3 of us here on the panel, we probably differ by about 1,000 ribosomal genes. So we all have varying numbers of ribosomal genes because of massive duplications that happen all the time. And in fact, they're more common. Duplications are almost, if not more common than single nucleotide mutations. So they happen all the time. And then if they subfunctionalize, then they end up becoming linked together. And then you end up having a genome-wide effect on traits, right? And what that enables selection to do is pick through individual variants and favor whichever one ends up being the adaptive trait. That's what the infinitesimal shows. And that's a sort of quick biochemical way to try to explain how that might come about.
Thank you, Dr. Zach Hancock. Paul, floor is yours. If there's anything you'd like to add or respond to, go ahead.
Yeah, I mean, polygenic traits are exactly, the type of thing that creates this problem. It's basically, as my opponent stated, it's the princess and the nucleotide paradox, because, individual changes to individual alleles often, in fact, most of the time, do not create any phenotypic change. This is called mutational robustness. And the literature actually testifies to the fact that there is a strong connection between complexity, organismal complexity, and mutational robustness. So that's the princess and the the princess and the nucleotide paradox. And this is why genetic entropy is unavoidable. This is the reason. The infinitesimal model does not overturn Kondrashov's paradox whatsoever. It just solidifies the need to realize that it is indeed the death knell of neo-Darwinism.
Thank you, Paul. Zach, you get the last word. Go ahead.
Yeah, that's just totally wrong. Like the infinitesimal completely buries Kondrashov's model. That's what Nick Barton wrote in 2013, or that's what Brian Charlesworth wrote in 2013. That's also what Nick Barton wrote in 2020, who I cited for the infinitesimal model. They both point out the fact that if most genes or most traits are underlied by many genes, and those genes are all effectively neutral, then the infinitesimal model applies, and it absolutely overcomes it. If you don't believe me, then you have to explain how it is that we've been able to select for things like corn oil when the vast majority of the genes contributing to it are all effectively neutral. How can selection still manage to work? The infinitesimal model explains it beautifully. We've seen it both in nature as well as in agricultural settings. If you can explain that, then it seems like genetic entropy is not a problem if most traits are polygenic.
Thank you, Zach. Next one comes in from Redefine Living. Thank you very much for your question. Question is for Dr. Hancock. Redefine Living says, AI criticizes your work by overusing compensatory mutations. How would you respond to this criticism?
That if you ask AI nicely, it'll tell you anything you want to hear.
Short and sweet. Paul, anything you'd like to add there?
I mean, that is true. If you ask AI nicely, it will tell you about whatever you want it to tell you.
Okay.
Yeah, the compensatory mutations, I'm not sure exactly what is meant by that. And I also don't see, I mean, I think basically what it might be saying is what I said, potentially. I'm just guessing here, but it's basically saying there are too many beneficials compared to deleterious. And I agree with that.
Zach, anything you'd like to respond to based on Paul's comments there?
I mean, there's not, right? There's an overwhelming number of deleterious, and their average effects are like you get far more deleterious, and the average effects of the beneficials are 0.001. In fact, we showed that the highest beneficial effect was 1%, right? That's the largest one that we saw in the entire simulation was 1%. The most of them were teeny tiny. So I don't know where that comes from.
Thank you, gentlemen. This one came in early on in the debate, but I'd be curious as to your thoughts. If it was addressed, feel free to reiterate the point, because I think reiterating some of these points on a technical topic like this is helpful to the audience and myself. So Creation Myths says, Kimura specifically removed selectable mutations from his calculations because he was modeling drift. I mean, seriously, Kimura said that he did that. It is more so directed at you, of course. Paul, what are your thoughts?
I mean, it's so silly to make this kind of a claim. Kimura put forth the model because he thought it was realistic. The idea that scientists write papers that they know are misleading and unrealistic is interesting to me. I don't think creationists could get away with that, and that's certainly not what Kimura meant. And as I stated in my original talk, it's not the case that Kimura removed selectable mutations from his calculations. At an effective population size of 1,000, Kimura's DFE has 52% effectively neutral, which means conversely, it would have 48% selectable. So no, they're not missing. And what he said in the paper about including beneficial mutations was kind of a silly little aside that he put in there. Yeah, if you put in an unrealistic proportion of beneficial mutations, they will overbalance and it will create a Darwinian demon or a Hancockian demon, which is what we have in this situation. So don't do that. Use a realistic ratio, and then you don't get the demons. I don't want demons in my sim.
Zach, over to you.
The entire concept of a beneficial mutation in the absence of any ecological context doesn't even make any sense. And this is why we talk about Darwinian demons, and why most simulations don't actually use beneficial mutations is because they cause the populations to explode and they blow the simulation up. And also because beneficial mutations are not expected to just be constant. Like, you expect beneficial mutations bring you to a fitness peak and then no more beneficial mutations occur. You're following around the fitness peak. It's called compensatory mutations. That's what the theory shows. That's what the literature shows. I'm also a little confused about your statement about Kimura here. Kimura modeled effectively neutral, the slightly advantageous mutations, and he gave a distribution for them. He didn't give a proportion, he gave a distribution. You can put the numbers in and like calculate it like whatever number you want, right? He didn't actually say it's this proportion. He just gave the numbers, and then he showed the relationship. And he said that if for 4NS, for NES equal to or greater, or for greater than 1, then you have K is equal to 4N V sub A. So basically that the rate of molecular evolution becomes equal to the population, or becomes proportional to the population size, which is really large, and it's not what you see in nature, right? So the argument is that when you include beneficial mutations, often these models, they don't work well, they break down.
Paul, over to you.
Yeah, it's true. He didn't include a proportion because that is entirely dependent on the population size. It's a function of population size. And that's why I gave that parameter. His DFE gives you 52% effectively neutral at a population size of 1,000, and that is not a realistic DFE. The realistic DFE comes from Rossimo and Schreiber, or at least it's much closer. I'm not going to hang my hat on that one paper as if it's the be-all, end-all, but it certainly is far closer than any of these other ones that I've seen. And under that one, basically, it would agree with my opponent. Nearly all mutations will be effectively neutral. They will be under the drift barrier.
Appreciate it, Paul and Zach. Next one here comes in for Paul. Laborious Cretin. Does he know genetic recombination fixes bad flaws also? Besides being able to pass along better or worse parts, mammalian cloning of clones generation cap from flaws accumulated. Paul, any thoughts?
He's saying recombination fixes flaws. I guess in the sense that it can prevent you from inheriting a deleterious mutation. It doesn't fix the flaw. It means that you didn't inherit it. But the problem is we don't have enough recombination in the real world we don't have free recombination. Now, that is one of the assumptions of this type of model, which is why we need an effective population size much smaller than census to compensate for the fact that we don't have totally free recombination. We have linkage blocks. So, yeah, but it's not quite clear what he's trying to claim here. If he thinks recombination fixes this problem, I would suggest he should put the realistic recombination rate into SLIM and see if it fixes the problem. That's what I did, and it doesn't. So, yeah, It doesn't. It's objectively, it doesn't do that.
Thank you, Paul. Zach, over to you.
I think he's talking about a recent paper in which I believe it was mice, in which they basically cloned them over successive generations, and those populations went extinct, basically. And the argument of the paper was that, well, what you've done is you've removed recombination because you're cloning individuals, right? There's no more sex. You basically removed sex from the population. And then when you do that, the population goes extinct. And so the argument then is that, well, if you can keep mice in labs for a long time, then sex must be benefiting their survivability, right? Because recombination is basically breaking up associations between good and bad alleles, and that enables selection to actually differentiate between them. And that's how you can, one of the ways of resolving many of these, like Muller's ratchet and stuff like that, is by basically having recombination breaking up association. So I assume that's what he's referring to.
Thank you, Zach. Paul, over to you if you'd like to address
Yeah,
those.
Dr. Joel Duff put out an article touting that paper, and that paper is nothing that would refute GE. Dr. Sanford would readily agree that recombination helps slow down genetic entropy. Cloning would greatly speed it up, just like inbreeding greatly speeds it up. When you inbreed, you're more likely to be homozygous. You're not getting the recombination effect. And the same is true with cloning. It would greatly speed up the decline. But the claim that is being attached to that is that somehow this means that recombination can rescue a population from GE. That claim is certainly not supported by that mouse paper. It's not supported by anything I've seen at all. And if you think it is, again, use SLIM. That's what my opponent used. Put the realistic recombination rates in. Let's see the proof in the pudding. Let's see it work. It didn't work when I did it.
Thank you, Paul, for that final word there. We've got a follow-up here for Dr. Hancock from Examining Origins. $20 super chat. Thank you very much, Rebecca, for the support and also question for the debate. Dr. Hancock, you mentioned that this model is evidenced by all the food we enjoy eating. Can you explain?
Yes, I would love to. Okay, I'm going to introduce you to a guy you've never heard of. His name is Jay Lush. Jay Lush was an animal geneticist from the 1930s, and he was greatly inspired by Fisher's work. Fisher was really a theoretician. He didn't do a lot of like application, right? But Jay Lush realized that if you take Fisher's model and you actually make a quantitative breeding program based on what he called breeding values, and today we call that additive genetic variance, then you can actually set up a breeding program that enables you to know exactly how to breed things like the amount of milk yield that is expected from a bull. Bulls don't give milk, but using the breeding value of the bull, you can actually estimate how much milk its offspring is going to have. And this is due to the regression approach that Fisher used. So he basically went all around the world, convinced farmers, livestock breeders to use this model, and then they applied it. And if you look at the rate at which crops have improved over the past century since we've applied that model, it's exponential. Like you look at chickens, for example, like the growth of chickens over the past century has been wild. They're now like 4 times larger than they ever were. They mature faster than they ever did. It used to take 120 days for a chicken to go to market. Now it's less than a month, right? So like that is the power of the infinitesimal model applied to the food that you eat. And I want to remind everyone, it assumes all underlying variation is effectively neutral.
Thank you, Zach. Paul, over to you.
Yeah, I mean, I wish I could say more about it, but this isn't something I've really studied in depth. It doesn't have anything to do with anything that is related to this debate, as far as I can tell. It certainly doesn't have anything to do with the simulation that my opponent used in his published work that he claimed invalidated GE. So it's kind of a non sequitur as far as I can see. It's certainly not an overturning of GE.
Dr. Hancock, you get the last word if you'd like it.
I mean, Brian Charlesworth 10, 15 years ago would disagree, as would Nick Barton, who would disagree as well. Both of them have published on this and shown, again, that the genetic variance is what is equal to the load in a polygenic model. And it completely resolves both Kondrashaw's paradox as well as Haldane's paradox. And I also want to stress that neither of those things are actually observed empirically, right? Like these are mathematical conceptions that when you go out in nature, they're called paradoxes because they don't actually seem to reflect anything real, right? We don't see them happening in nature. I gave you the infinitesimal model that makes actual predictions about nature, both natural things and in agricultural settings. My model works in nature. Put your model to nature's test.
Thank you, Zach. Great Q&A so far. Creation myths is coming at you again here. Paul, perhaps we could reiterate or expand, which could be helpful to myself and the audience here. Creation myths. This was obviously at some point during the debate. He caught up and he argued that Zach covered the big point, saying Paul admitted that the underlying premise of genetic entropy is wrong. Paul, what are your thoughts?
Yeah, Dr. Dan is always very good at making sweeping claims, but that claim is evidence-free. I don't know what he's referring to. I don't know what admission I supposedly made that the underlying premise of GE is wrong. I certainly didn't. So without further elaboration, it's hard to respond to this because I don't know what he's referring to.
Zach, perhaps you'd like to expand a
Yeah,
little?
what he's referring to is the underlying premise of GE, like going all the way back to Sanford, is that there's a zone of no selection, where he literally calls it the zone of no selection. And he's equating that to effective neutrality. Anything that's effectively neutral is in the zone of no selection. However, from the cross-exam alone, we showed that actually Paul is able to pick out which one is slightly beneficial, which one is slightly deleterious. So selection is doing the same thing. It does still operate at that level. It's just at the individual generation, the variance is dictated by drift. But if you take a step back and you look at the whole range of the distribution, they are biased, and selection is the cause of that bias. So the premise of GE that effectively neutral mutations are going to overwhelm selection is fundamentally wrong if selection is actually still capable of biasing distributions, as the infinitesimal showed, as the papers that I cited showed that it could, and as Paul himself agreed that he could differentiate those distributions.
Thank you, Zach. Creation myths clarifies, and then we'll hand it to you, Paul, for a response. He says the premise being that nearly neutral mutations are unselectable. That is the central claim of Sanford's book and model.
Okay, yeah. Using the term unselectable is a convenient way to basically communicate without going into the detail of saying, yes, it is biased, but not enough to overwhelm selection. I'm afraid that both Dr. Dan and Dr. Hancock here are actually strawmanning not just me and not just Dr. Sanford, but they're even strawmanning people like Dr. Kondrashov and Dr. Lynch, because no, it is not the case that the zone of no selection means that selection isn't acting at all. That is not the case. It is the case that the zone of no selection means that that's the cutoff point where drift overwhelms what little effect selection is having. So yeah, there's a very slight bias, but that bias is negligible in the long run, and it isn't enough to overcome drift. And again, I don't think SLIM even has a drift barrier. So this is irrelevant to what I did with the sim. It's irrelevant to what they did in their sim, because technically speaking, it's more of just a smooth gradient. And the drift barrier, if you look at what Kondrashov did, he put it in one place at 1/4 NE. But Kimura put it in a different place, 1/2 NE. And other people like Irewalker and Kitely just say NES is much less than 1. So it's not one specific drift barrier. It's just a conceptual idea to simplify an understanding that as you go down toward 0, drift progressively starts to overwhelm selection. And that is what is being modeled in SLIM. There's not a discrete drift barrier in SLIM, but you can see that the end result is still genetic entropy, because genetic entropy does not depend upon a discrete drift barrier, and it does not depend upon the claim that effectively neutral mutations have selection coefficients of 0. That's simply not, that's a straw man.
Okay, thank you, Paul. Since this was a new or follow-up point to the previous point, let's do one final round on this issue. So there's a few points there that Paul made that I'm sure Zach would like to respond to. Then we'll wrap it up because these were the last comments that came in, and we're doing pretty good timing-wise for Q&A. So Zach, over to you if there's anything you'd like to respond to there.
Yeah, it feels a little doublespeak to say that the zone of no selection doesn't actually mean no selection. It's no in quote in scare quotes. So like that doesn't seem very fair. It's like you're kicking the goalpost down, right? And it seems very clear from Sanford's writing that he means no selection, that selection cannot act on these things. And I'll also clarify the reason that you see different values on whether it's 1 on 2n or 4n. It depends on whether you're basing it on just drift, which is 1 2n, or whether you're basing it on the probability of fixation, which would then be 4n. It's a factor of 2 errors are very common in PubChem. I guess what I would like to drive home is that it seems to me like Sanford meant no selection at all. And that was the way in which he pitched his model, was that everything would be below the realm of selection, whereas effectively neutral mutations actually are selectable. They do get selected. And if there are lots of them, then you can very clearly distinguish their distributions.
Paul, floor is yours again.
I mean, I don't think we need to necessarily resolve what was in the mind of Dr. Sanford when he wrote his book. Did he mean to say absolutely no selection? I don't think so, because again, he understood the difference between effective neutrality and strict neutrality. Strict neutrality would be a selection coefficient of 0, and effective neutrality would not be 0. It would be a positive number, but that number would fall below a drift barrier, whether it's 1 over 2n or 1 over 4n. It falls below a drift barrier, which makes it negligible compared to the effective drift. And that's what SLIM is modeling.
Thank you, Paul. There is a question of clarification for you, Zach. So perhaps we'll get Wesley Coleman's question in here. Thank you, Wesley. Plus, again, thank you to the chat. We've had some good, solid interaction tonight and a good mix of viewpoints as well. Is the argument from Zach, that selection accounts for all mutations, therefore they did account for neutral mutations in their model? Correct me again if I'm wrong. Zach, over to you.
I'm not sure I understand what he means. Are you talking about in our paper model or in the model I presented today? That's, I guess, what I'm unclear on. Selection does not account for all mutations, even in our model. In fact, there were a lot of deleterious mutations that fixed in our model. It's just more beneficials fixed. So that's the distinction there. So there were also no truly neutral models. We modeled our distribution like Paul did with a gamma, and so there's no actual 0 there. So everything is, now you can have truly neutral mutations in SLIM. You just model a different mutational type and you set its effect to 0, right? And then you can basically have neutral mutations. But we had no truly neutral mutations in our model. I think that's what he's asking.
Thank you, Zach. Wesley is in the chat. So as we hand it to Paul, Wesley, if you wanted to clarify anything, feel free to do so before we wrap up here. Paul, anything you'd like to add?
I mean, I also didn't entirely understand the question, but I do think what I will comment on is that it's interesting. My opponent has made a number of videos in the past, as I stated, and a number of arguments against GE. And tonight he's made the decision to ignore everything that he's ever said or published and come with a completely different claim and basically deny the model that he used in his published work, which I find very bizarre that he would not want to defend his own model or his own sim. But that's what we've seen here tonight.
And Zach, you get the last word. Go ahead.
Yeah, I mean, I like to mix it up. I don't know. I thought it would be fun to talk about something new. Also, like, I don't have to defend my own sim. For one, you presented a simulation that I didn't get to see beforehand, right? You ran a bunch of simulations. I don't know how you parameterize fitness. I don't know how you parameterize selection. I don't know what you did, right? You got to spend time figuring out what I did. I didn't get to spend time figuring out what you did. So I can't offer a critique of your model. But then I also gave you another series of papers that investigated near neutrality in the exact same way that Dan and I did. So we did nothing novel. I've tried to stress that in the paper and through here. Our model is exactly that of Tomoka Oda's, is exactly that of Dan Hartle's. And they came to the exact same conclusion that we did. The reason that I wanted to use the model of the infinitesimal is I was hoping we would get out of the world of discussing abstract math and simulations and be able to talk about real biology, right? Because the infinitesimal applies to real biology, not to just simulations on your computer. That's why I wanted to talk about it in that manner, because it has actually been applied in real systems, whereas your simulation and Kondrashov's math has not. And they don't stand up well in real systems.
Thank you, Zach. A question of clarification here from Jackson Rowe. He's wondering, is it Stanford's zone of minimal selection in light of what you guys were going back and forth on, or no selection?
Who is this for?
That's for you, Paul.
Yeah, I mean, I defend what Stanford called it the no selection zone because it is effectively no selection. It's not literally no, but again, it's selection in an amount which is negligible compared to the effect of drift. This is what everybody has been explaining all the way back to Kimura. Kimura didn't say no selection. He said the drift barrier is the place where drift begins to overwhelm the effect of selection. Not that there's zero selection. Everybody going back to Kimura has been clear about this, and Stanford cites Kimura and Kondrashov. Stanford is not manifestly not saying anything different about that zone than what Kondrashov and Kimura said about it. You know, the fact that he calls it no selection is just, again, it's a way to simplify this for a lay audience. So if you want to call it zone of minimal selection, by all means do that. But it doesn't change the result of what we see happening, which is fitness decline.
Thank you, Paul. Zach, anything you'd like to add to there?
I mean, it's obviously false that it's negligible with respect to drift, right? I showed you the distributions of effective neutrality, and you could pick them out. If they were negligible with respect to drift, then all of those distributions should have looked exactly the same. But they didn't look exactly the same. And in fact, when Kimura himself modeled slightly advantageous mutations, they looked very different than what you would expect under drift. And whenever you look at the probabilities of fixation of slightly beneficial, slightly deleterious versus truly neutral, where drift is the only thing dominating, you see differences, right? A slightly advantageous of the same effect size is 4 times more likely to fix than a neutral, 54 times more likely to fix than a deleterious of the same effect size. Selection is still absolutely acting. As you already admitted at the very beginning, you need much fewer beneficial mutations to compensate given the distributions that I showed you.
Thank you, Zach. Paul, you get the last word if you'd like it.
Yeah, that's just not correct. The probability of fixation for anything that is below the drift barrier, and you can go to Dr. Michael Lynch's work on this, it's simply equal to the frequency of the mutation. So that means it's 1 over 2n is the probability of fixation. There's going to be a slight bias away from that, but again, that is a negligible bias. It's essentially going to be 1 over 2n. It's going to be equal to the frequency, and that's why the rate of fixation cumulatively over time for effectively neutral mutations is equal to the per generation mutation rate. So I explained that. And by the way, Donnie, could I ask you to please put that link to GitHub in the description of this video? Because I see people are asking for that.
Yes, and I was going to put up that question next, actually.
And we do have that link. I provided it. Let me copy it into the chat here.
Yeah, and then I can put it in the...
Because I definitely want to be transparent about that. I don't want people to think that I'm not showing my work here. Let me see.
And also, for those that are watching this after the fact, you did flash the slim code on the screen earlier, so they are free to screenshot it.
The slim code is included here in this GitHub.
Okay. There's another final question that came in here. Let me just find it. Chat's moving quick. Here we go. Common one. BioSteve17. Mice have much faster mutation accumulation than humans do, due to their incredibly short generations. Why haven't they gone extinct? For Paul.
Yeah, that's not correct. He's needs to check his homework on that one. Mice have, because they have short generations, that means there's less time for the germline mutations to happen between generations. Mice have a lower germline mutation rate per generation compared to humans, and it's significantly lower. So he's got that exactly upside down there.
Thank you, Paul. Dr. Hancock, go ahead.
Yeah, it is true that mice do have a lower mutation rate. It's not, I don't know if it's significantly lower. It is lower, but I don't know if it's that much lower. And I think it would definitely be, given their generation time and given, I guess, the time since the flood, if that's what we're comparing, then there's definitely been enough generations for us to have expected to see considerable decline in the fitness of mice. At least it should have been measurable. We should see like extensive genetic decay to some level at this point. Mice should not be reproducing as well as they would have been under the model. And in fact, you know how to use Slim now, so you can actually code up a mice model and you can test it for yourself.
Thank you, Zach. Paul, final word.
I actually thought about doing that, ironically enough, but I ran out of time. I've been running Slim for at least 6 months nonstop on my computer. I'm afraid I'm going to blow, the cores of my CPU. So I think I'm going to give it a rest for a while. But this is certainly something someone could try. You will find that it's a lot easier to find data on human parameters compared to other creatures, because humans are more, studied. I mean, obviously mice are heavily studied, and I'm not saying you couldn't do this either. And I would recommend someone trying this to see what Slim does. And I would also say that I don't necessarily disagree that mice are less fit on average today than they would have been, 2,000, 4,000, whatever years ago. I'm not denying that. Mice are, they're not as large and multicellular as we are, but they are still, what I would call large multicellular eukaryotes. So I'm not making the claim at all that mice are somehow immune to genetic entropy, but you got to realize that mice have massive population sizes and undergo a lot of selection, more than humans, obviously. And at large population sizes, the fraction of effectively neutral goes down. So that in itself means that there's more selection happening in mice compared to humans. So I don't think it's a problem for GE at all.
How would you handle this pushback from Creation Myths? He says, okay, so if mice are not declining, that means genetic entropy is fake, just straight up. What are your thoughts, Paul?
I didn't say they're not declining. I said they're not declining as quickly as humans because they have shorter generation times with lower per generation mutation rates and much higher effective population sizes. So again, if you want to try to put this into Slim with accurate parameters, go ahead, and maybe we can see when we should expect mice to go extinct. I don't know the answer, but I'm not claiming they're not declining. I just don't expect them to decline as quickly as humans.
Thank you, Paul. Zach, floor is yours.
Yeah, I want to spend a moment talking about this comparison between human and mice population sizes. Paul seems to think that effective population size is some like fixed thing that like humans are 10,000 and that's just what we are. The reason that humans have an effective size, an inbreeding effective size of 10,000 is because historically our populations were much, much smaller in size. But as we have grown in size, the effective size of the population will gradually reach the census size of our population. That is inevitable. That equilibrium will inevitably be reached. Now, if you then take the human population size at like 8 billion today, then the zone of no selection ends up being extremely small. That's why I picked that 7 billion number, is because, again, the human population size is really large. The effective size will eventually grow to meet it. The distinction between those is really the relationship between genetic diversity and population size, right? And at mutation drift equilibrium, then it is, genetic diversity is for NA of mu. And so eventually you'll reach that, you'll have that standing diversity, and selection will be just as strong, in fact, much stronger than it is in mice, because mice do not have effective population sizes of 8 billion. I can tell you that.
Thank you, Zach. Paul, last word if you'd like it.
Yeah, Dr. Hancock's claim that effective population size is going to equal at some point in the future the census size, that's demonstrably just completely wrong. It's a misunderstanding of effective population size. Historical population size is one aspect of the calculation, but it is only one aspect. Another aspect would be, the assumption of random mating, which is inherent to these models. And if you're going to correctly parameterize your model, you have to account for the fact that mating is not, in fact, random. And the way you do that is to lower the effective population size significantly lower than census. This is what everybody agrees. Nobody is using an effective population size anywhere near census. You can also go to the work of Dr. Michael Lynch, and I cited his slides actually in this debate. And one of his slides actually explains the effective population sizes of various different types of creatures. I believe he puts vertebrates in around somewhere between a range between 50,000 and 100,000 is where he puts vertebrates. So to claim that the effective population size is ever going to be correctly viewed to be census size is just completely against the literature and totally wrong. I can't believe he's making that claim.
Okay, to be completely fair, we are close to 50 minutes for the Q&A. It's been a great Q&A. I've really enjoyed this, and it's largely because we've had an excellent chat with several people who know this topic quite well. So it helps to make this debate that much more thorough in its content. Because I can tell Zach probably wants to respond to a couple things there. Let's go one final round, and then we're going to wrap up tonight's debate. Zach, let's go, let's say 30 seconds each. So 30 seconds to a minute from Zach, 30 seconds to a minute from Paul, and then we will conclude the Q&A. Go ahead.
Paul's understanding of effective population size is laughably bad. It's remarkably bad. So random mating is an assumption that Paul doesn't even really understand. It's about spatial structure. And spatial structure actually acts to inflate the global effective size. So it lowers the effective size locally and it inflates it globally. This is literally what I do for a living, by the way. I make models about effective sizes in spatial contexts. So there are lots of things that reduce the effective population size below the census size. That's why I picked 7 billion instead of 8 billion. But actually global NE is inflated whenever you have spatial structure. That's the whole point, because it takes longer for drift to happen. And N of E is in effect a measure of drift. That's what the effective population size is, right? And so if drift takes longer because you're all spread out, then the effective size is larger. That's why spatial structure actually acts to inflate the effective population size. And I'll stop there.
Thank you, Zach. Paul, go ahead.
Yeah, I didn't have time to wind up showing this slide in my presentation. I'm going to show it right now. One slide from Dr. Lynch's slide deck here, his Population Genetics 101. Do you mind if I show that one slide?
Sure, just let me know when you're ready for it. Let me put it up now.
Yeah. Let's see, share screen.
It looks like it's already there.
Okay, yeah. So here it is. So genetic hitchhiking via selective sweeps causes the genetic effective size of a species to be much smaller than actual census size. So this is called Hill-Robertson interference. It's also called linkage disequilibrium. But this situation here, the fact that you have linkage blocks is another one of the elements that causes effective population size. In Dr. Lynch's words, must be much, underlined the word much, smaller than the actual census size. So yes, I'm sorry, Dr. Hancock, your statements simply are not accurate here.
Okay, gentlemen, that was an excellent debate. I really enjoyed myself. I'll definitely be digging into the contents of this debate some more. I'll be rewatching it just to be able to fully absorb all of the points and arguments made on both sides. To the live chat, I appreciate all the comments, challenges, objections, and questions that came in. Those really help to make this debate on genetic entropy in many ways, specifically the question, are mutational effects a problem for evolution, that much more comprehensive and also enjoyable, especially for those of us that enjoy a good technical debate. So, okay, gentlemen, you guys are both good sports. Clearly, you have put in the necessary time, work, energy, and effort to know the topic well enough to have a good, profitable debate on it. So let's get some final words, final thoughts. I appreciate, again, the time not only for the debate, but also the time leading up to the debate, the work that you both put into it. So, Paul, let's start with you. Thanks again for doing this.
Yeah, and thanks again for having me. Thanks again to my opponent, Dr. Hancock. Like I said at the beginning, my criticisms are not in hatred in any way. They are in love. I want to see everyone to come to the knowledge of the truth. So, but that's what these debates are for. Again, I commend my opponent, Dr. Hancock, for being willing to engage in this debate, which is something most of his peers would not have done. So he deserves all the credit for that. And I thank both of you for your time tonight. Thank you for hosting this debate, Donnie. And thank you to God for enabling us to do this. This is part of God's plan for us to debate and to come closer to the knowledge of the truth. So that's what I hope happens here.
Thank you, Paul, for those final words, final thoughts. I also have your channel and relevant links in the description box of this video, and they've been posted throughout the night in the live chat for people who want to see more from you. Dr. Zach B. Hancock, thanks again for joining us here on Standing for Truth for another great and edifying debate. So, Zach, over to you. Final words, final thoughts.
Yeah, I had a ton of fun. I appreciate that Paul actually took the time to figure out SLIM. This is a thing that I've asked, creationists to do for a long time, and I really respect that he's spent the time to do that. I appreciate that, and actually dig into our model. This is a thing that Dan and I asked people to do, and he took the time to do that, and that deserves some kudos on his part. So I really appreciate that, and it made for a better discussion because of it. So thanks to that. Thanks to everyone for being here. I hope you learned something interesting. Yeah, really enjoyed the time here. Thanks to both of you and everybody here.
Dr. Hancock, thank you for those final words and final thoughts. Same with you. For those that want to see more from Zach, I do have his channel linked in the description box of this video, and also it's been posted throughout the live chat. Okay, well, again, you guys are good sports. I respect anybody who's willing to get outside their comfort zone, enter the debate coliseum for an exchange like this. I think it's important that we get out of our echo chambers once in a while and engage those of differing viewpoints. And we've got several more of those kinds of debates coming up as well to look forward to. So, okay, it looks like there's an after show right after this over on Creation Myths channel where the conversation will continue. So that's good. Paul, Zach, thanks for doing this. To the audience, I hope you enjoyed this and had as much fun as I did. Thanks again for your encouraging words, comments, super chats, support, and contribution for tonight. God bless you all. Standing for Truth is signing off.